Good morning. Can everybody hear me? So I want to welcome everybody to the first opening grand rounds for lab medicine and pathology. And also, this particular grand rounds is one of the 4 grand rounds that will be dedicated to the Division of Molecular Pathology and Genomics. And we're very pleased and really privileged to have with us today Dr. Todd Druley from Washington University in St. Louis. And I first met Todd at Children's Oncology Group, where we're both part of the Myeloid Disease Committee. And really, I can only say that he is really an amazing individual, a terrific colleague, extremely collaborative in nature, very generous with his expertise, with information. He collaborates actually with several individuals within our department and within the medical school, with Barat, with Logan Spector. And I've learned a lot just from our conversations with Todd. He's also a terrific communicator, and I think that's pretty evident in the way that he has set up his website, which I guess he got some help from a graduate student in his lab. So Todd, by way of kind of history, did his medical training at the University of Illinois, and then went to WashU for his residency and his fellowship in Pediatrics and Hematology Oncology. And he's now faculty there and also directs the fellowship program there as well in Hemonc. He is extremely well supported grant-wise, and his research, actually, he's made some major contributions to 3 major areas of research that you can see here. And again, as a physician scientist, all of these have both basic and translational components to them. So the first one in infantile leukemia, particularly in leukemias that have KMT2A, or what we used to know as MLL rearrangements. He's doing some really very forward-thinking types of research now in that area, using CRISPR technology and making patient-specific iPS cell lines. He has a clinic that's devoted to pediatric cancer predisposition. I think that WashU was one of the first places that recognized that patients with Beckwith-Wiedemann syndrome, initially actually many of them coming from assisted reproductive techniques in terms of pregnancies and the issues there around epigenetics, but now, again, much work that's expanded beyond those overgrowth syndromes. And then the one on your farthest right on sequencing technology, and that he's actually going to talk about today, although he told me that he's prepared to talk about any 3 of these, so that if anybody really wanted to hear about something else, that he would just pull up that talk. I think that another thing about Todd is, for some of us that we can really learn from, is if I really want to know what's going on in his lab plus in the areas in which he works, I just need really to go to his Twitter, which, as most of us still have our phone and fax number on our contact information, Todd has his Twitter feed. And so by looking, you can see just some major breakthroughs in research that are relevant to the areas in which he studies. So with that, again, I'm just going to turn over the podium to Todd and let him talk to us today about his new areas of research in sequencing and minimal residual disease.
Great, thank you. All right, well, thanks very much. Let's see, the thing to advance the slides, or just the buttons? All right. So, yeah, it's really a privilege to be here. I've had a lot of great collaborators over the years from University of Minnesota. One of my early chances, the infantile leukemia project that Betsy mentioned, was presented to me originally by Julie Ross, and tragic loss when she's no longer with us. But that work continues, and it was an idea and an opportunity that she gave me 7-8 years ago. And I've enjoyed working on that with Logan Spector, and Amy Linneberry and now Aaron Marcotte, and so many good people there. I've known Bharat for many years. We have a few different collaborations together, and I always enjoy picking his brain on what's coming next in the arena of clinical and molecular diagnostics. And Betsy and I have been kicking around different ideas for collaborative work together for several years, so hopefully we'll be able to pull something together here pretty soon. So it's a real pleasure to be here. I really enjoy this university and the friendly people that I meet here every time. So I was going to talk today about the third option on the list, which was the sequencing technology. And so one of the things that my lab has tried to do is improve next generation sequencing so that we can really use it as a tool for pediatric cancer. I'm a pediatric oncologist by training, and so you may or may not know there's flow cytometry to look for residual leukemia in a child following treatment, and this is known as minimal residual disease testing. And flow cytometry has really advanced and improved our ability to find children who have not responded appropriately to chemotherapy and may need additional treatment. But flow cytometry has its limitations. It's excellent for lymphoblastic leukemia because you have a clonal T or B cell surface rearrangement. It's less efficacious for myeloid leukemias because there isn't typically a single clonal cell surface marker. There can be a constellation of different markers that create a surface immunophenotype. But it's been shown multiple times that those surface immunophenotypes can change from the time of diagnosis through therapy. And so the limits of detection for flow cytometry have been a little bit less positive for myeloid leukemias. And in adults, myeloid leukemia is the most common malignancy, 25% survival, not the most common malignancy, the most common leukemia. With a 25% survival rate in children, the survival rate is about 60-65%. So much better, but I think we can do better. And flow cytometry only gives you a binary output. You get a yes or a no. There's disease there, there isn't. And it doesn't tell you much about what you could use to potentially treat that resistant leukemia. So we wanted to come up with a sequencing technology that could match the sensitivity of flow cytometry. So for lymphoblastic leukemia, it has a sensitivity of about 1 in 10,000 cells. It's about 1 in 1,000 is the threshold for myeloid leukemias. And then be able to not only just tell you if there's disease or not disease, but by looking at a panel of different genes, we could hopefully provide some insight to the clinician on targeted or precision therapies that may be effective for that particular patient. So that's the background of what I'm going to talk about. There's no disclosures. So, when you're looking for something that's 1 in 10,000, right, that's, really the needle in a haystack analogy. And so I have a lot of people tell me, well, that problem's easily solved. Just get a magnet and you've solved that problem. So what if the problem's even more complex than that? You're looking for a single needle in a needle stack, not a haystack. So now you have to change the way you're thinking. And so I'm going to switch a little bit here and just give you the background of where these very rare events currently stand in the field of developmental hematology and leukemia. So this was published a few years ago in the New England Journal from Harvard. This is Ben Ebert's group. So they looked at about 17,000 people that provided blood for a diabetes study in Harvard. And so they thought, well, if we look at the blood, we should be able to see clonal mutations in the blood that are associated with AML. And so by doing that, now they're just using standard exome sequencing here, nothing else. They're finding an age-dependent increase in the amount of clonal hematopoiesis that individuals have. And so in this sort of age group here, 50s to 70s, you've got 5 to 10% of people that they can detect clonal hematopoiesis in genes that are associated with the development of AML later in life. Now, it wasn't just a random distribution of genes that were mutated. There was a huge overrepresentation of DNMT3A, which has been associated with AML since many, many years, I think. And then TET2, ASXL1, and then sort of a tail of different genes. So these people didn't have AML, and so this was known to be CHIP, for clonal hematopoiesis of indeterminate potential. And it was strictly defined as having one of these mutations of greater than or equal to 2% allele frequency. And there's really nothing biological about that 2%. It's because of the error rate. Sorry, I'm getting my slides mixed up. It's because of the error rate of the sequencing technology, which I'll show you in a second. So back to minimal residual disease. So this is the pediatric data on minimal residual disease. And so multi-parameter flow cytometry just means we have to look at multiple different surface immunophenotypes to be able to detect the leukemia population from normal developing myeloid populations. And you can see here that there's obviously a significant improvement for children that do not have any residual disease detectable by this technology versus those that do. But we still have a third of children who have no detectable residual disease by flow cytometry, and they still relapse and suffer inferior outcomes. So this is the sequencing problem that I was telling you before. So these concepts hopefully will sort of come together here. So these are, if you just did standard next generation sequencing, it has a relatively high error rate. It makes errors at about 0.5 to 1%. And you don't care about that if you're sequencing a single individual and you're getting 40 billion reads. Who cares? You can eat that in a heartbeat. But if you're looking for something that's much, much more rare, that's what skews your ability to detect that. So here we have a true mutation in P53, two different ones. And all the other black dots in these plots are noise that's introduced by the sequencing platform itself. So, you know, some people cheat and they say, well, I know I'm looking for this mutation because it was in the cancer at diagnosis, and so if I just see it there, then I can say it's positive and sort of ignore all of this. But you can't use that strategy for detection of new mutations that may either be selected for or introduced during treatment. So there's a lot of ways to do the error correction. It's becoming pretty commonplace. There's multiple different systems. Haloplex is molecular inversion probes. Archer has their core AML product. SafeSeq, a lot of people use duplex sequencing. That's out of Johns Hopkins. That's very successful, but it's not the most efficient in my opinion. Rolling circle sequencing, and so these are all DNA strategies, and then there's a new group of RNA-based strategies to do the same thing to try and improve the error rate. So I don't know if you've seen it, but earlier this year, Foundation Medicine published a paper where they had summarized all of the sequencing that they'd done on DNA and RNA from all of the cancer patients that they'd looked at least in pediatrics, and compared it to some adult data. So they had found that there's a significant increase in the amount of RNA variation in children's cancer compared to DNA variation, which is more common in adult cancer. They both have both, but if you're going to tally one or the other, the children are going to have many more fusions or aberrant splice products, things in the RNA that typically haven't gone discovered. So, you know, if we're thinking about how can we develop a minimal residual disease strategy, there's definitely not going to be one size fits all. Or you can't do one thing for DNA structural variation, RNA fusions, point mutations. You just can't. So, you know, we want to think about this as sort of a toolbox. How can we improve the sensitivity of the sequencing technology, but still be sensitive to different types of genetic variation that we might encounter per patient? So this is the way that we've done it, and I think other people are doing similar types of things. So you simply use the mutation in your cancer as a biomarker, and by adding this UMI, so the unique molecular index, so every DNA molecule, this is different than indexing individuals, where you put 10 individuals in a sequencing lane and each one has their own index. This is putting a random index on every DNA molecule from a single person. So as you put a different index on these template molecules, they get copied through PCR and through the sequencer, and errors will be introduced in that process at random, which are the yellow boxes here. But the true mutation is fixed at the same position in every read that shares the same index, right? So if you didn't have the index here, you might think, well, this is a common mutation in this patient, and these are more rare mutations in this patient. Think about the plot with all the black dots on it. But in actuality, when you line them up, because you know they all came from the same template, you can say, well, those aren't actually true mutations. That's just noise introduced by the sequencer. And this read, or these reads from this template molecule have totally different mutations. And so you can align all of these reads with each index against each other, and then discard the noise and retain the true mutations. And so the student that did my website figured this out too. He was furious with the prior website, so I said, make a new one. So then this that I showed you before becomes this, okay, because you've just tossed out all of that noise. And so it's easy to see these mutations that would be indistinguishable from noise previously. Now we can see them quite readily. And they stay fixed at their relative locations, and the noise level drops much closer to baseline. So we've done some serial dilutions with this technology. So this is for Down syndrome work that we've done, but it's also nice because GATA1's in the X chromosome. So here's a 4-base insertion. Here's an X chromosome mutation. So across 5 orders of magnitude. So we're getting down to 1 in 10,000 molecules in a heterogeneous DNA sample where we can detect the mutation. And we do technical replicates at each time point. These are 2 other known mutations. And so we feel pretty good finding them. We've done blood and marrow. We're still trying to get some spinal fluid to look for that too, but it should be conceptually the same. So we feel pretty good about the limit of detection. We're in the range where we can do similar things to flow cytometry. So we've published this a couple times. So the first time we got to do this was with Tim Lay and Rick Wilson, Dan Link. So their question was, Dan Link's been studying therapy-related AML for a long time. So about 2 to 10% of people that get treated for any type of cancer will go on to develop myeloid leukemia months or years later. And so many of those cases have p53 mutations. And so the question that Dan Link wanted to ask was, are these cases due to mutations that were introduced by the chemotherapy given for whatever primary malignancy, or was it selection for a pre-existing mutation in the hematopoietic compartment? And so he had had a lot of foresight to collect blood from patients that had been diagnosed with any type of cancer for the last 20 years. And then he had done whole genome sequencing on their therapy-related myeloid leukemia, picked out the cases that had p53 mutations, and then asked us if we could use this sequencing strategy to go back to those pre-existing blood samples and find the p53 mutations. So the punchline there is that, yes, the p53 mutations pre-existed, and about 40% of healthy adults have p53 mutations that are non-synonymous. And it's simply, if you think about it, you give myeloablative chemotherapy, the bone marrow compartment is fairly well eradicated, but the cells that are resistant to apoptosis and have some sort of growth advantage are going to be the ones that come back. And so we were able to detect these p53 mutations as much as 12 years before the patients actually presented to their oncologist with myeloid leukemia. So that was our first publication with it, and then Andrew published more of this is more of just a paper on the technology, how to do it. All the sequences are in there, everything that we couldn't put in the prior paper. So that was good because we knew what we were looking for ahead of time, and that was great. But we don't want to just be looking for the same things we already know about. We want to have a strategy that matches the sensitivity, but is also able to detect new mutations. And so to do that, we need a panel. And it would be cost prohibitive to try and do this with an exome or anything of that scale. So instead of us having to build our own myeloid panel, we just pulled the Illumina panel off the shelf. This is their TrueSight myeloid sequencing panel. It sequences parts or all of 54 different genes that are most frequently mutated in myeloid leukemias. It covers about 141,000 base pairs. But at the time, Illumina didn't offer any error correction. And because we're not that clever, we just wanted to make this as plug and play with Illumina as possible. We do Illumina sequencing, so you could just take the adapters out of the box that they mail to you and put the adapters, sequences that we published. You order them from IDT, just put them in, do the protocol. There's one extra quantitative step, but other than that, it's just the same as what you're already doing for Illumina sequencing. So unlike some of the other strategies that have completely different molecular biology strategies, this is pretty plug and play. And so we use this because now we can try to discover new mutations across this whole panel of different myeloid-related genes. This is the gene list of what it looks like on that particular panel. Now these are adult genes. There's very, very few genes in this panel that are going to be relevant to pediatric AML. I just want to point that out. So the point here was to we've spent a lot more time trying to quantify what normal physiology looks like for mutations that are this rare. And so before we could say that's something that's disease-related, we had to figure out what's actually just normal. The idea was after finding that 40% of healthy adults have P53 mutations, they're never going to get cancer or leukemias, so we better quantify what's real, what's physiology first. So this has been an interesting collaboration with the Nurses' Health Study. So this is out of Harvard, and they started collecting blood from nurses back in the mid-1970s. And the reason for the nurses is because they were able to draw their own blood with no consent and just send it in. So they've agreed to send in a vial of their own blood every 10 years that they're on the study. So they've got 125,000 nurses that are enrolled in this study, and they've got blood samples every 10 years. And so we went to them and we said the study that we really want to do is we want to compare the clonal mutations in the blood samples from nurses that stayed healthy versus nurses that got AML. And they told us they didn't know what we were talking about. There's no way the sample's 40 years old would have that kind of detection. And so they said, just show us that you're not going to screw things up first. So we were able to get healthy nurses first because they've got a lot of those, and so it actually was the right thing to do because it was a completely unexpected result. So we got 20 healthy nurses. We got 2 samples from each one. So they've been followed for 30, 40 years after their first blood draw. They get annual checkups. They know they're doing well. And so the mean age of collection was 57 and 68 for the second collection. Now we validate everything with droplet digital PCR, which you can't really use for discovery, but it's excellent if you know what you're looking for, and I'll show you some of the validations. So the punchline of all this was that here's those other papers. So this is the Jeswell paper from Ben Ebert's lab. This is a paper out of WashU. I can't remember where the Genovese paper comes from, but around age 60, late 50s, they're reporting that 5% or so of people have clonal mutations that you can detect. We actually found, if you use a more sensitive technology, 95%, so 19 out of the 20 nurses had mutations in their blood in genes that are AML associated. So it's happening to everybody. And what I didn't expect to see was the exact same pattern that they saw using the less sensitive standard exome sequencing. I thought what we would see using a strategy that's 100 times more sensitive than just standard exome sequencing. And I will say, I forgot to say this earlier, just deep sequencing doesn't cut it, right? So if you're just going to say, we'll just do deep sequencing, that helps you detect a mutation that's present at, say, 15, 10%, maybe down to 5%. But no matter how much sequencing you do, you're still generating noise at the same rate. So if you want to get below the noise threshold, just deep sequencing itself is not going to cut it. So just a lot of people don't really appreciate that difference. But at any rate, I figured we would just see kind of a flat line of just stochastic mutation across all of these different genes. And that's not at all what we saw. We saw the exact same pattern. So over 40% of the mutations that we found were in DNMT3A. TED2 was the second one. ASXL1 was the fourth. And so, and we still can't really explain this, right? So you think, well, if this is happening, there must be some positive selection for DNMT3A mutations. Before I get to that, here's the validation. So this goes across 2 orders of magnitude from 10% to less than 1 in 1,000. And you can see by droplet digital PCR, we're pretty accurate across the entire spectrum. So I think we're doing pretty well. So back to the selection question. Remember, we've got 2 blood samples from these healthy nurses. So if there's positive selection in the nurses that have the DNMT3A mutations, you should see that clone expanding 10 years later. And we don't really see that. I mean, so here's technical replicates of time point 1, technical replicates of time point 2, and each line is a different mutation, each box is a different nurse. And so, you know, this has gone from, you know, I mean, this is maybe 10 times greater, I guess, so you could say that particular mutation has expanded, but there's another clone in there that hasn't. These clones haven't really done much, a little bit of expansion there. I mean, this was only detected in the second time point, nothing here. So it's not like there's such a positive selection pressure that these are just growing exponentially in these people. And so then the next question was, well, maybe you're not actually detecting the hematopoietic progenitors, you're detecting long-lived B or T cells that just float in circulation for some period of time. So we were able to flow sort these old blood samples and just separate them into B, T, and myeloid cells. That was the best we could do with the quality of these samples. And compare that to the bulk measurement, and there's not a whole lot of difference between them. You know, I mean, this scale only goes up to 1%, so these increases may look dramatic from the back, but this is going from, you know, 0.5% to 1%. And so, you know, it seems as it suggests that the mutations, because they're in all 3 of these different blood cell compartments, are actually in the hematopoietic progenitors, and they're just cycling at a pretty steady rate without much difference in the background. So then we were able to actually get permission to do the study we wanted to do up front, and then so we can say if everyone has these rare AML clones in their blood as a function of age, then why do so few people actually get AML? So now we've got 105 people, 35 people that actually got AML later in life, and they were matched 2 to 1 for individuals that stayed healthy. So they were matched by age, gender, and ethnicity. And so that's a total of 105. And now we were blinded, so the Nurses Health Study just sent us 105 samples, so they're in trios. So we know the trios. We know in every trio there's 1 case and 2 controls, but we don't know who the case is. And so we want to know, are there specific mutations that are associated with developing AML later in life, different combinations of mutations? What's going to be the differences in these folks? So this is the total number of mutations between cases on the left and controls on the right. Now that isn't a huge difference. I mean, there's slightly more mutations in the people that get AML, but not a ton. You know, this is an average of 4%. This is 7%. So, you know, it may be significant. It may not. We validated everything under 1%. Just those are the most rare ones, and we did pretty well there by droplet digital PCR. So not a tremendous amount of change, but if you look out of 141,000 different nucleotides that we sequenced to quite significant depth with error correction, only 3 residues actually associated in any way with the development of AML. And again, we have a relatively small sample size, so that may be the problem. But the 882 mutation, the arginine 882, is well known for being associated with AML. And it actually acts like a dominant negative. So DNMT3A has to homodimerize to execute its de novo methylation function. And so when you have an allele with this mutation, they still homodimerize, but it inactivates the complex so that it cannot perform its function. Many of the other mutations seem to either don't bind at all with the wild type, which would just be sort of inert, or they bind and don't have any dysfunction on the methylation capability of the complex. Now, ASXL1 and JAK2, so the JAK2 is interesting because it was only found in males, so there's a separate study, the Health Professionals Follow-Up Study, that was started about 10 years after the Nurses' Health Study, that includes people from EMTs and lab techs, things like that. So it had some men in there, so we could compare men against the ladies. Now these aren't quite significant, but because the confidence interval here does cross 1. But the odds ratios, this isn't an odds ratio of 1.3 or 1.4. These are strong odds ratios, and this is based on a conditional analysis. So again, it's a small sample size. It's not going to be rock solid, but I think you can infer that there's definitely some association with the development of AML later in life. So I would actually argue that as some of these panels get to be more and more and more and more, that may not be the right strategy, because just having a DNMT3A mutation doesn't really mean much for your risk of getting AML. So these lollipop plots, this is the protein sequence of DNMT3A. These are all the mutations we found in the cases. These are all the mutations we found in the controls. Right? So here's 882. So there's 8 in the cases, but there's 4 in the controls, 2 for the H change and 2 for the C change. Now here's this, I know you can't read this, but this is ASXL1. So and it's pretty similar across both. In JAK2, the only mutation that was found at all was 617, and there was 1 in the controls. So I'm of the opinion now that this has to be solved by a single-cell strategy. So if we have a healthy person that has, say, 10 subclonal mutations and a person with AML has the same number, what we are insensitive to here is the fact that 4 or 5 of the mutations may be in a single cell in the person that gets AML, and they could be completely spread across 10 different cells in the healthy person. Yeah?
Did you look at copy variation at all? You'd expect that as the DNA evolves and becomes more unstable, then maybe there'd be copy number variation.
Yeah, that's a great point. We haven't done that because we don't get a ton of sample, and they're not the greatest quality from 1977. But that's a good point. It may actually be a difference. Any other questions? That was a great point. Could be the answer. Where we want to take this next is to try and see how many of these are co-localizing, and maybe that's going to help give us the answer. So this is very difficult to read, but so this is for each gene, right? And so you see the genes up here, and the cases are in 1, so there's a little 1 and a 2 under each of these. So you can compare the percentage of mutated alleles between cases and controls, and I'll just point out a few things. So here's DNMT3A and TET2. Really no difference between the amount of mutated alleles in the cases versus the controls for either one of these. But here, we start to see genes. Now, there's so few mutations overall that they don't come out in the conditional analysis. But you see big disparities between the cases and the controls for ASXL1, B-core, RAD21, there's a big difference here. GATA2 is only found in the controls, none in the cases. IDH2. Again, going back to the single cell idea, I'm thinking that you may see a DNMT3A mutated clone or a TET2 mutated clone, And then it has one of these secondary, tertiary, quaternary mutations inside the same cell. But again, maybe not. There could be different mechanisms involved. So that's a study that we're hoping to get out pretty soon. I mentioned Down syndrome before. I'll show you a little bit of data here. So this is a new COG study, 1531. The PIs are Hans Hitzler from Sick Kids in Toronto and Jason Berman from Dalhousie in Nova Scotia. And so the point here is that children with Down syndrome AML have never had a particularly robust minimal residual disease strategy. Their cells don't have really particular immunophenotypes that historically have been detectable. So most of the mutations in these kids fall in exon 2 of GATA1, which is on the X chromosome, right? And a few of these in exon 3 at the very 5' end. So what we did is, instead of using a whole panel, we just developed 2 amplicons, about 300 base pairs that would encompass the entire exon 2 and then the front part of exon 3. And so we're going through these cases prospectively to see what we can find. And again, I apologize for the difficulty to read this, but the point is that we can find that 83% of the cases have GATA1 clonal mutations at diagnosis that we could then use to track for residual disease. And so some of these we've got end of induction 1. So here's a case where you've got 21% at end of induction, at diagnosis, and then less than 0.3% after a month of chemotherapy. Here we've gone from 75% to 5%, so I would expect this patient should still be positive. This should be an induction failure, right? And here, there's 2 different mutations at diagnosis, but neither one was detected after a month of therapy. So, we expected this to be more like 95, 96, 97%, so why only 83%? And as we've gone through, we've looked at other things. So we found one patient has a 445 base pair deletion across GATA1. So that's why we couldn't PCR amplify, because the primer sites had actually just been completely deleted. Some of these others we've run through the bigger panel. And while we're not seeing a lot of GATA1, here's an intronic mutation, but we see other mutations, particularly in spliceosome genes, that were reported by this Japanese group a few years back as being associated with Down syndrome AML. So maybe there's completely non-GATA1 mechanisms. Anyway, we're still kind of digging into that, and that's been fun. So back to the more structural variation. So everything I've shown you now is either point mutations or very small indels. So one of the things that we're not sensitive to with the strategy I showed you before is internal tandem duplications of FLT3, which is important. And if you're not familiar with this is a receptor tyrosine kinase. And frequently you can get duplications in this juxtamembrane domain, but they vary significantly in sizes. They can be just a few bases, they can be hundreds of bases. So that presents a very different computational problem. So what we've done work with Archer in their core AML. So they have a little bit different strategy that's worked very nicely, and we've been able to adopt this. So what we've done previously that I showed you up to now is we have one primer that sits on either side of the target on the same strand, so we get strand-specific amplification. We fill in a cross between the two, and then each of those primers has a universal PCR to amplify from that strand, right? And then you take that and you align it back to a fixed reference sequence. Now, you can think how internal tandem duplications are not going to be very robust to that strategy, because you could have a little duplication, you could have a huge duplication if you're forcing it back to a fixed reference. A lot of the aligners just don't like that. There's too many mismatches, and you have to do it on sort of a case-by-case basis to try and weed through this actual reads and do it that way. So this is a little bit different strategy. So on either side of your target sequence, you can have 3 or 4 different primers, and then 3 or 4 different primers the other way. So you can make 9, 16 different sized amplicons, maybe more than that. And then instead of forcing those to fit a fixed reference sequence, you just align them locally to each other. So you do a local de novo alignment. And then you can take all of those reads and figure out exactly what the size is, because you're not assuming that they're fitting a fixed size. So when we do that, we get nice, very robust data. So here we can see 71.7% of the reads had a duplication of exon 14, but had a starting point here and a different starting point over there. 17.4% of the reads were wild type. There was no duplication. And we had a different start site here. So the remaining 10.9% of reads had the same exon 14 duplication, but we had a different starting point. So these come from a different template molecule, right? And so this is the same. So we can say, you know, there's 80+% of these reads have the internal tandem duplication of that nature. And so we've been able to, I didn't show it here, but we've been able to take a cell line that we know has a fixed internal tandem duplication and do the same serial dilutions and match a sensitivity of 1, or a limit of detection of 1 in 1,000. So we feel pretty good at 1 in 1,000. We can't go below that with this platform. So that's all DNA. Now I'll finish up a little bit here with some RNA work. So if you just do standard RNA-seq, you know, this is just one example of how, as you get to lower and lower transcript numbers, the correlations start to fall off between your sequencing number and your concentrations. So you lose accuracy as you have lower and lower amounts of transcript. So if we put the UMI onto RNA molecules, and this is just using the QIAGEN Human Cancer Transcriptome Kit, but QIAGEN sells it without UMIs. We put the UMIs on there. And we're doing technical replicates from different samples. We're getting exceedingly accurate transcript counts down to very, very rare. I mean, these are in single digits. So when we do droplet digital PCR, our sequencing found 4 copies, ddPCR found 7. You know, so it's not perfect, but it's pretty darn good. It's definitely better than bulk RNA-seq. And you can see that our ddPCR across multiple orders of magnitude lines up pretty well. So you can use the UMI strategy to gain accuracy at very, very low transcript counts. And I think that's important because everybody's very excited about single-cell transcriptome sequencing, but all of the transcriptome single-cell strategies are typically only getting the top 20 or 25% of RNA molecules that the cell has. So you get a pretty broad dropout of more rare things. I know a lot of people who spend money and time doing single-cell RNA transcriptomes, and then they're frustrated that they're not seeing their gene of interest. And it's not because it's not there, it's that it's not being transcribed at a high enough amount to be picked up by the beads or whatever the enrichment strategy is. So you can either do a huge amount of sampling to try and overcome that, which adds cost and time. You know, again, this is not a single-cell technology. I'm not trying to bill it that way, but it's highly sensitive for transcripts that are rare within your cell population. So another thing we're finding is that non-coding mutations, particularly around splice sites, can create multiple different funky isoforms in different patients, or in a single patient. So this is a patient who didn't have a coding mutation, right? They had a non-coding mutation, but it was near a splice site. And so they get multiple different isoforms of RUNX1 that don't fit any known isoform assemblies that have been described. So here we have exon 2, 4, 4, and 5. That was in 35.6% of the reads. 26.4 have exons 3, 4, and 6. You know, we've got 6 or 7 examples of this. This is just a model, but so you can have a non-coding mutation. A lot of people say, well, there's no coding mutation. It's probably irrelevant. And then if you look in the RNA space, you can say, well, actually, that could have huge implications. I don't know if these are translated to protein, so that would be the next step for this. But this is just an observation that we've made that I think bears some follow-up. So using that strategy, and again, you don't have to read all this, but we looked at, so the target project for AML looked at several hundred kids through the Children's Oncology group from the prior de novo phase 3 study. And so we've taken 76 of those, looked at the RNA from those end of induction 2 samples, so they've finished 2 months of chemotherapy and should be in a full remission. And so by the COG's reports to us, only one of these patients had a known fusion. But you can see here, so these are the 3 children that died during therapy. Here's all the relapses. And then here's the ones that stayed in remission, at least at the last point of being measured. And you can see that 65% of the time we're finding AML-related fusions that remain in these patients even after 2 months of therapy. And so again, you can see that there's a high percentage of the CDFB-MYH11 in both samples. This is early days, so we have to figure out what the thresholds would be. Why would it be okay in these patients but not okay in these? Maybe it's not related. I don't know. But we're finding these things as we look, and I think it just changes sort of our frame of reference for what's meaningful and what isn't. This is some work that we've done with Andy Kolb and Aaron Crougy. So Andy's the director of the Myeloid Disease Committee in COG. And again, just more busy, but 67% of cases, these are at diagnosis, have at least one fusion. So you can see all the different fusions here. They're different allele frequencies, whether it's ALL or AML. So we're following these through. They've actually done xenografts with a lot of these, so then we can follow the expansion of the xenografts in mice and see if it's recapitulating the clonality of the primary diagnosis. So that's something we've been working on for a while, so that's been fun. But then, one thing we want to do is try to figure out how to tie all this together. I think the gene expression stuff gives us these nice little graphs that we might be able to use in the future. So each little panel is a gene. There's marks underneath that are for different isoforms, so there's only one isoform for these 3, multiple isoforms for NUP98 or some of these others. And then each row is a patient. So you can quickly look across here and see where patients stand out. We've got one patient who's very overexpressed for retinoic acid receptor compared to the others. I lost my mic. Oh, there it is. Here we've got overexpression of a particular isoform of NUP98 in this patient. This is an over or underexpression in a few cases of this gene compared to everybody else. So, I don't have clinical relevance for this yet, but I think these are the way you can start to tie these data together to figure out what's relevant and what isn't. So Aaron makes these hairballs, which everybody sort of groans and rolls their eyes. But I think the beauty of this is, again, saying we don't have to sequence everything. If we know what these networks are, we can hit 2 or 3 critical nodes in each of these networks and hopefully infer some data. Well, maybe there's a drug for a different gene within this network, but there's good rational sense that it may work in our patient who's got a mutation in a different part of the same network. And by doing this, again, early days, but we're able to cluster individuals by genotype in these different principal component analyses. So there's some projects that we're trying to do. So we've expanded the error correction profile with Illumina's help. So we're now adding in 40 genes from pediatric cases that were detected by Sohail Mushinshi in the Target AML project. It's about 200 kb. So we've got about 2,000 samples from the latest COG phase 3 study, which just ended earlier this year. That's AAML 1031. And so we're getting all 2,000 of those. And so we're going to do the error-corrected DNA and the error-corrected RNA on every sample. We can compare that to the flow cytometry that COG has on every sample. We know the outcomes for the patients. And so what we'd like to do is use some machine learning to try and build these different principal component or a t-SNE plot, something where we can take multiple dimensions and reduce it down and say, you know, kids that have DNA and RNA and flow cytometry, whatever, they don't have to be mutually exclusive. Let's use all the tools we have and figure out who's most at risk. Because if you're not aware, the current therapy for AML was established in 1973 and it hasn't really changed significantly since then. So hopefully we can start to do something better. And you know, so we have a lot to learn from this. I'm really excited about this study. It's taken me 2 years to get all the nuts and bolts together, but we're just about to get going. So with that, I'll stop. A lot of people to thank. And again, I appreciate being here. It's been fun to talk to Betsy. And yeah, I'll take any questions you guys have. Yes, sir.
By using the archer chemistry, how long ITD you were able to detect?
So that's a good point. So with the really small IDTs, we can still align to a fixed reference. But if it's above about 30 base pairs, we need to do the de novo alignment strategy. So we've been able to detect, I think the biggest one's 200 some, 240 in that range that we've found so far. You know, we're just sort of limited by what we have, but that's the biggest one. Yeah.
So Todd, have you looked at the Down syndrome patients who have just the transient myeloproliferative disorder, and then those who remit and versus those who go on to actually develop AML with respect to GATA1?
So Betsy's question pertains to Down syndrome. So a lot of these children are born with a myeloid expansion called transient myeloproliferative disorder, and that is thought to be due to myeloid expansion, which is occurring in the liver prenatally, but then as the myelopoiesis switches to the marrow, a lot of times those clones just die out. So, but then about 25% of those kids will go on to get AML, and it usually has the same mutation architecture as their transient myeloproliferative disease. So she's asking if we've compared and contrasted those. So as it turns out, the Children's Oncology group can be a bit Byzantine, and so some of those TMD samples are locked away in a freezer somewhere under heavy guard, and other samples from the matching Down syndrome AML patients are in a different freezer under even heavier guard. And so we've spent about 18 months working on permissions. We've got scientific council. We got everybody said yes, please give Todd these samples, and we still have no samples. So hopefully those will turn out, because that'll be a fascinating question. Yes, sir, Bharat. This will be good.
All right, Todd. I had a question about your the UMI barcodes. You use 16 base pair barcodes?
Yeah.
And you know, other people have used barcodes of different sizes. Is there a rationale to choose one particular size over other?
It's fairly arbitrary. I mean, we've tried 8, 12, and 16 bases. And so we felt that there was enough errors introduced during the synthesis of the adapters that if we went with an 8 base pair index, we couldn't be as certain of the different molecules actually being separate ones. There was just, between the sequencing noise and the synthesis errors, it just got a little muddy, and we didn't feel like we gained any. We actually lost sensitivity, and we didn't gain, you know, extra bases of sequencing or anything. And 12 was pretty much the same, so we just stuck with 16 because we can have a Hamming distance that's enough to where we can correct the synthesis errors and still have unique indices. But it's pretty arbitrary. There's not much difference. Yes, sir.
Have you or anyone looked at differences in the bone marrow stroma of patients that develop these leukemias? So these types of mutations in combination with changes in the activity of the stroma, could that be?
I think that's a huge avenue for future research. We haven't done it because we're getting samples that are, have been banked at other places for a long time. They're not actually our patients, but I would love to do that. So, in some of our other studies, we're trying to do xenografts, and so we, from the mice, we could do some xenograft work and then see if there's, evidence to support a role for stroma. I bet there will be, and I think that's probably pretty important. But we haven't done it. Bessie.
So since we have 5 minutes, I think, left, I'm going to ask you if you would give 2 minutes on what your cancer predisposition clinic is about, just so that people get an idea of what types of things are going on at WashU.
Sure. So about 4 years ago, I set up a clinic for pediatric cancer predisposition based on the idea that most of these children don't have an abundance of somatic mutation. And there's a lot of data suggesting that children, one of the things I'm particularly interested in scientifically is the link between abnormal childhood development and early childhood cancer. And I try to get people to think of it not as a disease caused by a ton of external mutations that have happened to you, at least not in children. Think of it as a developmental anomaly. If I tell a parent their child has a club foot or a ventricular septal defect or a seizure disorder, they don't think that that's due to magnetism that came from outside or their vaccinations, or at least I hope they don't. So, I want to think of cancer the same way. There's mutations there, but the idea is that mutations are happening stochastically and randomly to everybody all the time. And the mutation only matters if it falls in the right genetic context, at the right particular developmental stage. We know there's genes that are involved in adult cancers that have nothing to do with pediatric cancers and vice versa. So, recognizing that at our medical center we had a lot of children coming to see the geneticists, see the GI with polyposis syndrome, seeing neurologists for neurofibromatosis, or seeing whatever. But nobody followed them longitudinally for the development of cancer. And so we created a cancer predisposition program that, I like to refer to it as the hub of the wheel, because a lot of these kids need a lot of ancillary services, and so many of the parents, at least in our region, are pretty overwhelmed by what their children's medical needs are. So we want to take that burden off of them and be, the hub of their wheel. We can help them with testing and seeing other doctors, doing whatever they need to do, keeping up on the latest recommendations. So that clinic's been growing. Every year we're seeing, 50-60% more patients each year that we've been in existence, and so it's been a lot of fun. Roughly 10% of the kids will actually get a cancer. We've picked up a few Wilms tumors in kids just by screening abdominal ultrasounds in kids with hemihypertrophy or Beckwith-Wiedemann. We picked up some cancers in some of our Dicer 1 patients. Some of the research that we're doing, we just published a paper 2 weeks ago in the American Journal of Medical Genetics looking at how, this was a 10-year retrospective study from our institution. For every pediatric cancer diagnosis, what was the incidence of patients that we saw that also had a birth defect diagnosis? And that was defined by a group of people at our institution. And it turns out, if you look at the public data on how many kids have birth defects, it's in the 2 to 3% range, and 14 or 15% of our pediatric patients, over 1,200 patients, had a comorbid diagnosis of a birth defect. So there's good literature, epidemiology literature, saying that birth defects increase the risk of early childhood cancers, but these come from huge statewide or national studies, and they don't have a lot of resolution. So, we didn't have near the numbers, but we were able to show that kids that are born with a congenital defect in their central nervous system are more prone to getting brain tumors, but not any brain tumor. It's medulloblastomas and gliomas. All right, the astrocytomas, the ependymomas, there's no impact there whatsoever. And so, you know, again, it's helping us identify who is actually predisposed to potentially getting a cancer. And I think as a basic scientist, it starts to suggest that there could be commonalities between the abnormality of development, and this isn't just big structural birth defects, these are some movement disorders and also neurocognitive defects as well. So one of our nurse practitioners is doing a study on therapy needs. So compared to national data, the children that have needs for therapy is wildly significant compared to just a standard child with special needs. So it's a population that is growing in recognition, and I think makes for a good scientific query. We collect DNA from our entire nuclear families. So I want to know the proband. What did they inherit from mom? What did they inherit from dad? If they have healthy, full siblings, what do they have or don't have? And and then we bank their tumors. So, you know, we're trying to be as comprehensive as possible.
I want to thank you very much.
Thanks,